our research is focused on trying to reach and treat central nervous system disease and peripheral nervous system disease. And so what that means is and this is probably also gets into why I'm doing it, we're sort of pushing the boundaries of what medicine can do. So the great majority of our drugs available on the market, more than 98% of them, cannot reach the brain due to this biologic barrier called the blood-brain barrier. And so my group is really focused on how do we get these drugs and actually enable them to be therapeutic, because medicine exists that could work to help with a lot of these diseases. It just can't get into the brain. And so that's sort of the gap that my students and I try and fill, is how do we use chemistry, material science, polymers to create a system that can interact with and transport across the blood-brain barrier. And then we do the same thing in the peripheral nervous system, where there's a similarly difficult to breach barrier called the blood-nerve barrier. And this is why, in order to treat nerve diseases or nerve injuries, typically it's going to require a surgery. So they'll even, open you up to inject a drug that might work directly into your nerve. But we just published a paper with one of our invention disclosures with Kerf. We just published a paper showing that we can actually use muscular injections to travel directly to the nerve, so bypassing that blood-nerve barrier as well. We've got these polymer-based nanoparticle systems that are very versatile. So they can hold drugs. That's going to be our therapeutic delivery. They can hold like other nanoparticles or contrast agents. So that's our diagnostic delivery. Or because of the way the structure is set up, they can hold two things at once. So they can hold a drug at the same time as they hold a contrast agent. And that's theranostic delivery, which is therapeutic plus diagnostic. And so really we try and highlight and look into research areas at each of these sort of versatile spaces. So with therapeutic, we're delivering therapeutic proteins, therapeutic peptides, therapeutic RNAs. We can do that with our polymersomes, and that's a major thrust in our space. We also work simply on diagnostics. So actually we collaborate with Angela Alexander Bryant, who's Jesse's advisor, and we've been working to deliver gold nanoparticles inside of our polymersomes as a contrast agent. And we've just recently injected those into animals, and we see that it works very well. So we get very high contrast. And then we have a theranostic tool where we actually co-deliver proteins with iron oxide contrast agents. And that allows us to track where the nanoparticles go via MRI. And then as the nanoparticles release from the polymersomes, as the iron oxide nanoparticles leach out, the contrast behavior changes. And so it allows us to monitor where the protein is being delivered. And so that's sort of more of a theranostic tool. So we've got all of these different like plug and play pieces of machinery that we can work with. I'll start with the first. So the first is unique. It's actually not something I've talked about at all today. So it's not using a polymersome technology, but it was inspired by a collaboration that was started with polymersome technology. So hopefully I don't talk for too long, but bear with me. So I do a lot of work with Jeff Twist. He's at the University of South Carolina. He approached me as a baby 26-year-old professor after I gave a talk at a conference and said, hey, Jess, we want to try and get across the blood-nerve barrier. So he really pushed that whole thrust in my research group, which is now my first R01, like pretty amazing. But he had developed this technology that can regenerate nerves. And it's based on this whole internal biologic principle, which as a chemical engineer, I didn't know anything about, where when cells undergo stress, they pull away any mRNA that's not needed to translate survival proteins. So if it's mRNA for regenerating the nerve, that's not important for survival. The cell just needs to survive. And so what it does is it takes all these other proteins that are present in the cell and it binds them to the mRNA. And it creates this thing called a stress granule. And these stress granules are phase separated, but they're liquid. So they don't have membranes, they're just liquid droplets. but inside of these liquid droplets is mRNA. And he developed a peptide that can break this apart, great, and it allows for regeneration to occur. So we've been working with him to deliver this peptide. But always in the back of my mind, probably for the first 5 years of my career, was like, okay, I wonder if I could make that. I wonder if I could make a stress granule. Is there a way that I could hold mRNA in the cell? Like, let's say I want to hold this mRNA to make this protein, and I want to make it on demand. Could I keep it there in the cell for a long time, the way that stress granules could? And so at the time, an undergraduate researcher in my lab, Chloe Forenzo, she was thinking about what she wanted to do for grad school. She'd been working on a typical for my lab polymersome-based project. And I told her, I said, if you really want to stay here, you need to get a new experience. You can't stay working on the same project, as an undergrad. You need to transition to grad school and you need to start something new. And she was like, okay, I will. I promise I will. She canceled all of her other grad school visits. Bless. I'm like, you should have at least tried, but I'm so grateful she's here. And she stayed with me. And so finally I was like, okay, this student knows me, knows how I operate. They know the base technology, so they could always fall back on it if it doesn't work. That's a very important piece of mentoring a grad student. They have to graduate. So you need to know that something about their project is going to work. And so I said, she's the right student. I'm going to pitch her this crazy idea. And so I said, I want to make these liquid phase separated droplets. At the time, I didn't even know what they were called. Okay. They're called coacervates. I didn't even know. I was like, I want to do this to store mRNA for on-demand release. And she just rolled with it. So we started talking a lot about outer space. She's very interested in getting into outer space. She loves NASA. She loves space. And so our first sort of idea was like, well, could we give astronauts these mRNA vehicles? And when they reach a certain level of radiation, could we turn these proteins on? And these proteins are needed for radiation protection. And so that was her major thrust. It's like, okay, well, how do we make a material that can respond to radiation? Well, it turns out we just need to make it respond to radical oxygen species. And so the project's really taken on its own just major life since then. So, she's working on these ROS-sensitive coacervates for mRNA delivery in the body. And we were talking about, this system with some people on campus, just, loosely discussing, trying not to disclose too much. And Sarah Harcum in bioengineering said, Jess, you have an ROS sensor. You could detect ROS. And I was like, I didn't even think about that aspect of this technology. So she pulled me into AMBIC, which is the Biomanufacturing Innovation Center, and using the exact same technology. Now we're working with some people at AstraZeneca, Bristol Myers Squibb, and it looks like maybe Lonza's going to jump in with us, we hope, to use this technology to do something we didn't even think about doing, which is monitoring CHO cell stress and culture, which they can't do right now, but leads to a lot of loss of valuable product. So from this one tiny crazy idea, by understanding biology and wanting to make biomimetic materials, now we've got all of these different thrusts. Chloe is doing the NSF I-Core program, so she's got a lot of interest in, licensing this technology. I don't think we want to start a company, but we'll see. But we're like, maybe we could license it to other people and, then they could use it. And so that's the first tech transfer. And that's the first time I ever was like, do I need to be sending this out somewhere? Like, do I need to be protecting IP? So that was my first jump into Kerf. If you look out in the literature, which I just did, since we just published a paper on this, nobody's targeting peripheral nerves on purpose. It's very interesting, and I don't necessarily know why. But a lot more people are focused on how to get across the blood-brain barrier, and a lot less people are concerned about peripheral nerves. But when you think about the sheer number of people with sciatica that could benefit, or pain medication that can work directly on your nerve that doesn't require the surgical placement of a nerve block, we've got a lot of interest in this. So this was all with undergraduate students, which is also very cool. So an undergraduate student of mine, Kaylee Trumbull, she started working with me the summer after her freshman year. I added her to this project. We've been trying to work with Jeff to deliver this peptide. And I kind of told her, like, here's some different conjugation chemistry you can explore, but I want to try and come up with a nerve targeting peptide, and I want to test it in nerve injuries. And she, again, just kind of ran with it. She really thrived as an undergraduate student in that environment with that intellectual freedom. And then once we had developed the technology, we're ready to test it. We went to Jeff Twiss's lab at the University of South Carolina, and he taught us how to create sciatic nerve injuries in rats. And we started testing to see where our particles go after being injected intramuscularly. And what we found is that one of our targeting peptides allows for direct transport to the peripheral nerves, that where the drug is retained in the injury site for 48 hours at least. We haven't tested for longer than 48 hours. I think and hope that these technologies will be in clinical trials in the next 5 to 10 years. And I see this being a viable option. The biggest barrier is the scale up production of nanoparticles. So again, depends on what your knowledge is in that space, but it can be hard to make enough. But that's something we do really well. I'm a chemical engineer. So we have scale up procedures that we've already vetted that we know work. So I hope in 5 to 10 years we'll be in a clinical trial for the polymersome based treatment. I think what I hope for the coacervate-based technology is that we can get some interest from pharmaceutical companies who are willing to license our technology. What we think is that it could be used already in biomanufacturing. That would be a pretty easy integration. We're working to integrate that into a probe, which could monitor in real time, which would also be really cool. But we also think, based on what we've seen in the lab, is that these coacervated mRNAs could be encapsulated in nanoparticles. But what we think is that pharmaceutical companies would be most interested in doing this with their nanoparticles that they already have, that are already approved by the FDA, that have been vetted. And so we're hoping that we can license our technology to them, help work to integrate our technology into their nanoparticle platform. And then maybe in 15 to 20 years, we might see some of that in clinical trials. So one of my mentors in grad school, he's in clinical trials right now. And this is why I have a good sense of how long this can take. For my field, I feel like nanoparticles are moving into a personalized medicine space, just like all other personalized medicine. So, that's been a major focus in the cancer research space over sort of the past decade of every individual is not the same. They've got their own genetics. How do we fine tune the chemotherapy that's available on the market to address their concerns better? I think the same thing is going to start happening with nanoparticles. In the field and in the clinic, we have really created one nanoparticle system that we've applied to a couple of things. And I am of the belief, based on my scientific knowledge, that this, a lipid-based system is not going to solve every problem. It's got some innate concerns. They're leakier, so a lot of drug can get lost. They tend to go to the liver, so trying to get nanoparticles away from the liver. I think our field is going to have to move to a more personalized approach, actually looking at, okay, what is happening with this disease? And what system works the best for this disease, not a generic, let's just plug and play with, for example, just a lipid nanoparticle and just change this one piece. I think that could have some benefits, but I see us as a field, especially in academic space, where we're not really yet concerned about the making money piece. I see us pushing the boundaries and looking into, how do we design a very specific system for this very specific disease to get more of a personalized approach. Not necessarily a skill, but having had past leadership experience helps me to be able to actually run my team in an effective way, where everyone feels safe, heard, accountable. They feel like their contributions are valued, right? And so being able to have had that past experience in leadership and translate that to my lab now is one of the most helpful experiences that I had. Probably the most important skill, and I also don't know if this is technically a skill, but it's just a personality trait, is like humility. So science is hard. It doesn't work every time, and you're wrong a lot. Your assumptions were wrong. Maybe the way you interacted with someone was wrong, right? Being able to humble yourself, apologize, recognize the world doesn't revolve around you and other people have other things going on. It's also a really important, like, I will say that's a skill because you can train that, you can learn that to have. And it also allows like other people's voices to shine in our lab, because I maybe have one concept of an idea, maybe it's not the best one, but if I don't have the ability to be humble, I'm not going to hear the voices of my students that have a better idea or a better direction or alternative path that has the potential to be better. So yeah, maybe humility, leadership experience and humility. So the reason why I do what I do, and probably the reason why most professors do what they do, is because they love students, right? So it's just very amazing to see the growth and the trajectory and the confidence building that occurs specifically in this age demographic. Everyone's trying to figure out what it is that they want to do, and seeing things click and seeing passions being born, overnight or while I'm in the lab with my students it's just amazing and inspiring. Seeing them gain confidence in themselves is also very powerful. Them being rewarded or recognized for the hard work they put in. Yeah, it's the best. It's hands down the best part of the job.